So I'm going to be talking about some work that I've been doing over the course of the last year, essentially latent representation of the human epigenome. And I thought this would be a good time for me to come down to Stanford because I'm about to take my general exam where I have to present in front of my committee. So hopefully you'll be a softer audience than that, and you'll be able to give me valuable insight into how I can woo them, or at least which jokes fall flat. So I'm going to be talking about this. I'm going to be giving an introduction to what this means and why I chose Avocado. So hopefully that'll, as a name. So hopefully that'll all become clear by the end. So when the first draft of the human genome was published, it contained a long sequence of the 4 characters A, C, G, and T, which encoded for every base pair in the human genome just 2 bits of information, its nucleotide identity. This gave rise to a whole host of very important insights, such as the identification of regulatory elements that basically spanned for the next decade. Unfortunately, it was a very incomplete view of human genetics. An obvious problem that arises is how can we have the same human genome in each one of our different cell types, but have all of these different cell types? And there are many different cell types. You can see on the left-hand side here that in just an adult human, you have a large number of differentiated cells. You'll have lung cells, you'll have fat cells, you'll have blood cells. All of these are very different from each other, but they all arise from the same set of instructions. This gets exacerbated when you think not just do we have differentiated cells, but you have stem cells, undifferentiated cells, like those at the bottom here. You'll have stem cells from different stages of development, like fetal brain, fetal heart cells are very different from fully developed heart and brain cells. And so taking a view that we're going to understand all of these things just from these 2 bits of information per base pair seems like it would be a very difficult task. And so in order to kind of tackle this problem, these large nationwide consortiums were developed with the idea that we need more data. And so these 2 projects, the Encyclopedia of DNA Elements, or ENCODE project, which is ongoing, and the Roadmap Epigenomics Consortium, which completed in 2015, basically were devoted to the collection of data which we call epigenomic data, which is this data that measures phenomena that do change from one cell to another. So what they wanted to do is characterize chromatin state, basically characterize our genome by assaying as many different signals as possible. And what this means is that they were trying to quantify as many different phenomena that occur in the genome as possible. For example, they would run something like RNA-seq, and now it'll give you measurements of how transcribed various regions of the genome are. And a lot of the genome is actually transcribed. There are measurements like ChIP-seq, or chromatin immunoprecipitation, followed by sequencing. And this has 2 main categories. The first is to quantify things like histone modifications, and the second is to quantify things like the strength of transcription factor binding. Both of these are very important, and both of these give more information than just the 2 bits of information. Then lastly, you'll have things like DNA-seq or FAIR-seq, which seek to give information about how accessible a certain region in the genome is. Basically, how available are these nucleotides to the outside world. You can imagine that regions that are very accessible to the outside world are likely to be functional in that cell. That if you have a transcription factor binding site that is wrapped around a histone very tightly, it seems very unlikely that transcription factor will be able to bind them. Then lastly, there are more recent assays that have been developed to directly measure the 3-dimensional structure of the genome, like 5C, or more recently techniques like Hi-C or Hi-CHIP or GeoPet. And so all of these techniques are measuring different phenomena about the genome. And so these assays can be displayed basically like genome browser tracks. That what you have is some measurement for every position along the genome. And since these are sequencing-based technologies typically, what they end up being is the number of reads at each position in the genome. But it's not just the number of reads, because that incorporates a lot of biases like GC bias. And so you typically want to do a bunch of post-processing in order to get a more informative number. And the data which I'm going to be displaying throughout this talk is basically the log 10 p-value of a certain region being a peak. And what does a peak mean? Well, you can see, this region here, this histone modification H3K4ME3 forms some form of peak here. And H3K4ME3 is known to localize in promoter regions when that gene is transcribed. So perhaps these regions where it's peaking are promoters for genes that are transcribed in that cell. So now we have more information. But it's not sufficient to run each one of these assays once. You need to run each one of these assays in every cell type of interest. And so they want to try to characterize the epigenome by looking at as many different cell types as possible. The roadmap project looked at only 127 cell types in depth, whereas the ENCODE consortium is looking at many hundreds of cell types to try to characterize their differences. So there are many cell types, and it's going to be very difficult to look at each one of these and run all of the many, many assays that you would like. But it is important to do so because you get different information. For example, we looked at this track, and we saw that there might be some peaks here. But if we look at other stem cells, this is not cells that are very different from each other. You can see that while many patterns are the same across cell types, there are some patterns that are different. Like, for example, this peak here in E001 is not as strongly present in the other one. Or, for example, here it's much more strongly present in E003 than the other ones. And this variability from cell type to cell type is probably responsible for the diversity of these human cell types that we see. So we'd like to look at them some more. The roadmap project specifically looked at 127 human cell types, and they focused on these set of 24 assays. And in this matrix, where there are potentially 3,000 experiments that can be run, they only were able to perform around 1,000 of them. And you see you have the various human cell types of interest as columns, you have the assays as rows, and so there's this interesting block that forms right here. And that corresponds to the tier 1 and tier 2 cell types, which are very well characterized ones, like GM12878, H1, IMR90, so forth. Very well characterized cell types, very well understood ones. Then you have this block that spans all of the cell types. And this is because the roadmap project chose to take some assays that were maximally informative and run them in every cell type to see how that changed. And so these correspond to the assays like H3K4ME3, which localizes in active promoters, H3K4ME1, which localizes in enhancers, and others, and these other three. Basically, these were ones that were already determined to be incredibly informative to gene activity, and they wanted to do that. The problem is that there were other assays, and there are other assays that could be run that would provide other information, and it's likely impossible to totally fill out this matrix. And this matrix is just a tiny part of the total amount of information that can be done. So, have we characterized the human epigenome yet? That we've gone from having 2 bits of information, just the nucleotide identity of any base pair, to having these 1,014 measurements for every base pair, which is the signal value for some assay and some cell type. Does this give us all the information that we would want? Maybe, maybe not. And so there have been 2 primary ways to try to use this information to perhaps more fully quantify the human epigenome. Prior work has focused very heavily on saying, can we use the connections in the data that we have in order to impute all of the missing experiments? And so there are 2 main approaches. One is Chrome impute by Jason Ernst, who's at UCLA, and his advisor at the time, Manolis Kellis at MIT. And another one is predicted, which I'm going to talk about a little bit because it's developed by one of my co-authors and my advisor, and came out earlier this year. But we take another viewpoint. Instead of trying to characterize the epigenome by getting more measurements, going from 1,000 measurements now to 3,000 measurements, or 10,000 or 100,000 measurements, depending on the number of cell types and assays you'd like, we want to go in the opposite direction. We want to say, if we have these 1,000 measurements, we probably don't have 1,000 important independent phenomena that we want to study. We probably have far fewer. Can we learn an informative lower dimensional representation from the data that we have? And so this can kind of be viewed as like trying to distill the information that we have into this informative, into a more informative representation, saying something like, hey, this is a gene, versus saying, hey, marks that are associated with gene transcription are active and marks not associated with gene transcription aren't. So, this is very similar to a challenge from industry called the Netflix challenge. This is basically the canonical representation of how to do these types of factorization approaches. The idea is that what they have is when you watch a movie, you can try to rate it. You can give it, did I like this or not? But the majority of people like me don't actually rate the vast majority of shows that we watch, and we definitely don't watch everything on Netflix despite our best efforts. So you might end up with a matrix like this, but in fact, it'll probably be far sparser. Well, each one of your viewers is a column in this matrix. Each one of your shows is a different row. And then the blue boxes correspond to pairs where a rating exists. Not that the rating is good, just that there is a rating for that person. And if you have this very sparse matrix, how are you going to be able to identify what this viewer is going to like next? Because this was the point of the Netflix challenge. You want to recommend shows that a user hasn't watched yet that they would like. So how well would this viewer like this show that they haven't yet seen? And the approach they take is they want to try to build a profile for every user. They want to build a profile for every user. They want to build a profile for every show. And hopefully, these profiles will give you some insight into whether or not a viewer is going to like a show. The problem is that they can't just use your vector of ratings that you've given, because that's going to be incredibly variable length. What they want is this fixed dimensional representation on top that just gets more informative the more shows that you've rated. So what you end up with is these two quote-unquote skinny matrices of what are called latent factors. And each one of the columns here will correspond to the profile for a specific viewer, and each one of the rows here will correspond to a profile for a specific show. Each one of the latent factors captures some sort of informative property about either the show or the viewer. So you can imagine, like, the first column here might say, is this show thought-provoking? And the higher the value is, the more thought-provoking the show is, and the lower the value, the less thought-provoking it is. The corresponding first row in the viewers is, does this viewer like thinking? And at least for me, when I'm watching Netflix, I don't like thinking, so my value would probably be very low. Then you can imagine that the next phenomena is, does this show have Adam Sandler? Because it doesn't just need to be some abstract concept, it could be some very specific thing, like is a famous actor or actress in this show? And of course, the corresponding user representation would be, does this viewer lack standards? Because if both of those are true, then the viewer is probably going to give a much higher rating to the show, whereas if the viewer doesn't lack standards, then they'll probably give a lower rating. So, how do you make a prediction? Well, I say that these things were supposed to line up, and so what you can do is you can take the viewer representation, for example, perhaps my profile here, you can take a certain show's representation, like Gossip Girl, and what you do is you take the tail, basically the dot product between the two. You multiply the purple factor and add that to the multiplication of the blue factor and add to the multiplication of the green factor and so forth. And then hopefully, if it's a good model, you'll get that this viewer likes this show because I really like Gossip Girl. So then if you want to make imputations, all you need to do is just go through all of the show representations, all of the viewer representations, just substitute them in. So you go down from perhaps Gossip Girl Live, me and Gossip Girl Live right here. If I want to do the next show, then I just go down by one. And so perhaps this is Black Mirror. And then I'll get my imputed value for how I would view Black Mirror, then go down and maybe BoJack Horseman, and then just go down all the way, and for all viewers, and eventually what you get is this big matrix where you have all of the viewers, all of the ratings that actually exist in blue, all of the ratings that are predicted in purple, and then you can look at only the predicted ratings and say, I think the person's going to really like this show, so I'm going to recommend it. And that's the basis of their recommendation engine. So, the question naturally arises, that's great and all, but how do you get these representations? And I'm not going to talk too deeply about that, but the gist is, like most machine learning methods, what you can do is you can just frame this as an optimization problem. You can use canonical optimization techniques like stochastic gradient descent or the atom optimizer in a mini-batch setting where you try to train these until convergence. So, math, basically. And it's unlikely that the learning factors are going to correspond as neatly to the various things that I said, but they're probably going to correspond to rough ideas. Like, for example, they're probably unlikely to say, is Adam Sandler specifically in this show, but is a famous actor or actress in this show, and does this person seem to like famous actors or actresses? So, all we have to do is make a few simple modifications to this setting in order to apply it back to this epigenomics approach. That instead of having viewers and shows, what we might have is assays and cell types. And if we're modeling the roadmap compendium specifically, we have 24 assays, and we have 120 cell types that we'd like to model. And instead of it being mostly sparse and sporadic, as we saw before, it was very structured. The idea is that we have our tier 1 and tier 2 cell types up here. We have the very well-characterized assays, but the assays have been run in many cell types over there, and then we have very few of the others. Do you have a question? Okay. The other modification is that we don't have a matrix. What we have is a tensor, because like I said, we have these measurements at each position in the genome. So we have our assays like before, we have our cell types like before, but then we have this for every single position in the genome. We get these measurements for every single base pair in the genome, but it's frequently more, it's more common to analyze them at some aggregate, like 25 base pair resolution, because at a certain point you're just getting noise. Like most of these measurements of histomodifications really operate at the level of nucleosomes, so something like 250 base pairs or something. So if we model this at 25 base pair resolution, like prior work has, then we have 115 million positions that we like to model. So we can do basically the same thing. We can extend this matrix factorization approach that, epitomized by the Netflix challenge to have 3 dimensions. Now we have our genome factors that span the whole length of the genome, in addition to our cell type and assay factors. And we have the same, we can have the same gist, except with these questions corresponding to more like biological phenomena. Like, for example, is this a liver protein in some position? Is this gene encoded liver protein at some regions of the genome? If your assay measures gene transcription or gene expression, and your cell type is a liver, cell type, then you're likely to have a high value. But if any one of those is not true, like for example, if it is a liver protein gene and it is a liver cell, but this assay doesn't measure gene expression, then you're likely to have a lower value. And that kind of makes sense. So making a prediction is pretty much the same as with the Netflix challenge, except instead of making a dot product between 2 vectors, you're now doing a dot product between 3 vectors. You're taking whatever this first factor that you're learning for the genome position, multiplying it by the assay one, multiplying it by the cell type, then adding it to the multiplication of the orange and green and so forth, until you get your predicted value. And so this is called tensor factorization as opposed to matrix factorization. And so each value that you are predicting is now one single measurement in a single one of these assays. Each one of these experiments corresponds to a cell type assay pair, with this being the assays H3K4 ME3, and the cell type here is E001 or E002 or whatever. But your prediction is now one single value here. So it'll be that value here. And so what you have to do when you make these predictions is you have to go through every single value at every single position for each assay and each cell type, which can be kind of computationally expensive, but so this approach that I just described is the foundation of the method called predicted. The idea is that you learn these representations and then you impute these measurements, but predicted isn't just one tensor factorization, it's an ensemble of many tensor factorization approaches with something called a main effects model, which is a bit more complicated, and ultimately they're just able to impute this data fairly well, better than compute was able to, which was the other baseline that came out in 2015. So what I'm presenting on is our approach called Avocado, which kind of builds on this. So with the tensor factorization setting, what you have are these 3 sets of factors, and you use a dot product between them in order to get your prediction. The first way in which Avocado extends upon predicted is that you feed all of these factors into a neural network. And the reason that you would do this is, of course, because one of the best applications of deep learning is when you have lots of data and no real understanding for what's going on. You just apply a deep network to it, and you get good predictions. So, but on a more serious note, one of the real benefits of using a neural network here is that you can model these types of nonlinearities that exist across the 3 axes. That before you had to do this dot product, which was a very linear model, this gives you a nonlinear model. But additionally, you can change the number of factors that you learn for each one of the 3 dimensions. Previously, if you're doing this dot product, you have to have the same number of factors. Like in this example, there are 5 factors that represent a cell type, an assay, and a genomic position. Since we have 115 million positions, the vast majority of these parameters are going to come from the genome axis, so you want to make that as small as possible. but you don't necessarily want to limit your assay representation or your cell type representation. If what you're doing is concatenating these and feeding them into a neural network, you can set any number of factors for any of these axes and get a more informative cell type and assay representation while still constraining your genome one. The second way that we build up upon this is by considering the genome position again, by considering it and trying to constrain the number of factors that we learn. That again, if we have 1.2 million positions in just the encode pilot regions, which are a few chunks of the genome that were initially studied by the ENCODE project, then that's going to end up being 120 million parameters if we learn 100 position-specific factors, which was what was set up and predicted. That's a lot of parameters to learn. And if we're modeling each position in the genome independently, we're kind of ignoring the fact that all of these measurements are highly correlated. If you have a high mark in a promoter region, it's likely that the position next to you is going to be a high mark. If you're in a gene body and you're not measuring gene expression, it's likely that all of these positions are going to be low. It's not that each one of these are independent in a way that like cell types and assays are kind of independent. So we kind of resort to domain knowledge in order to reduce the parameterization here. Like I said before, that many of these phenomena that we're studying with like chip-seq data operate at the nucleosome level. So they're around 200, 250 base pairs. We have these big domains, like an entire promoter is likely to have very consistent signal. Entire gene body or quiescent region are going to span around like maybe 5,000 base pairs long. So what we do is that instead of just having each one of these parameters be independent from each other, is that we model our genome axis at 3 different resolutions. That we learn some factors that correspond to 25 base pair resolution, like these other ones, because that models things like DNA-seq very well. Then we have some factors that correspond to 250 base pair resolution, basically modeling a nucleosome. And then we have some factors that correspond to larger, perhaps like domain scale activity, like this is a gene body or enhancer or whatever. So this makes it go from 100. If we choose this, composition of parameters, well, we still get 100 numbers that represent each position. But instead of having 120 million parameters that we need to train, now we only have around 22 million parameters. This gives us around 6-fold reduction in the number of parameters without losing much expressivity. And it makes it a lot easier to train as well. So those are the it makes it much easier to train, and it reduces the requirement that we make this ensemble of methods like the predicted approach has, and we can just have a single unified method, which is important for a reason that I'll get to in a little bit. So now we have a model. We want to make specific decisions about the structure of the model, and I may kind of skim over this because I'm not sure how much time I have left. The idea, though, is that now we need to select the parameters for the model. How many factors are we going to use to represent the cell types? How many are we going to use to represent the assays in the genome? The actual dimensionality of any of these axes is unknown. We don't know how many dimensions should represent the cell types, and so we can't just plug that in from any type of domain knowledge. And then if we're using some sort of deep model, how many layers should we have? How many neurons per layer? There's a lot of questions about the number of hyperparameters we should be using. And so basically, the answer to this question is how much computing power do you have? That if you have more computing power, you can search through a wider space and try to find a better model. And so if you're in industry, this is great, because industry has lots of computing power. They have thousands of GPUs that they can just use to search forever, find the great model, and then be done with it. But if you're in academia, that's not so great. I'm very fortunate that my advisor has somehow acquired grant money to be able to purchase multiple GPUs that I can monopolize and be yelled at by the other grad students. But not everyone is as fortunate as me to have access to all of these. And so this is kind of like a mini side rant, which is that a lot of things that happen in machine learning right now are dominated by your computing power. And while computational biology for now is a little bit, is not targeted by big companies like Google and Facebook, this is a big problem that's happening in other fields like computer vision and natural language processing, that they will they won't have better ideas, but they'll have more computing power, and so can search for better hyperparameter settings. So I'm not looking forward to the day that Google decides that what they really want to do is genome science, because then I might be out of a job. So rant over. So basically what we did is that out of the potential tens of thousands of possible combinations of hyperparameter settings that we could have used, we ran 1,000. And what we saw is that many of these models were, garbage. That if we look at if we train on around 800 of the 1,000 tracks, validate on another 100 of the tracks, then just calculate our performance based on the mean squared error of the imputations that we're doing, then some of our models are worse than using compute. Many of our models are worse than using predicted, but some of them are very good. And so ultimately, we use the best model that performs on this validation set. So if we look at what hyperparameters seem to matter, a natural question is, well, did we even need to use this neural network? And the answer for this is clearly yes. That each one of these plots shows the mean squared error of all 1,000 models that we train, but looking at the settings for each one of these 7 hyperparameters that we cared about. And what you can see about is the number of neurons in each one of our layers, in each one of the layers for a neural network was the most important factor. And as we increase that, we seem to get better performance. Same with the number of layers, and there was some modding issue with Seaborn that I haven't figured out yet, so they just look like lines. But the best model is at the bottom here, so 2 neural network layers with 2,048 neurons. That performed the best, and so we chose to move forward with that. Ultimately, this isn't the most important part because we're trying to learn a distilled representation of the human epigenome, and so our factors are going to be what's important. But by using this neural network, we're able to learn factors that are more independent from each other than if we just use a linear model. So let's start looking at this representation that we learned. The first thing that we did is let's quantitatively take a look at the cell type embeddings. That we ended up using 32 factors in order to represent our cell types, and when we use TS&E to project this down in 2 dimensions, we get a very clear separation of cell types by their functional anatomy according to roadmap. This is particularly surprising because in the 2015 roadmap paper, they don't get anywhere near this nice separation by roadmap anatomy, and their approach to doing it is far more complex. They look at the correlation of various signals in the functional element of relevance, which is a bit more complicated than just saying, hey, we have these numbers and we're projecting it down. So you can see that the ones that kind of cluster the most distinctly are all the blood cells. And so all these blood cells are together, all of your GI cells are here, the stem cells are all here, both blue and green, or different types of stem cells, so them being close together makes sense. You have your brain cells over here. And it's kind of important to know that in the roadmap paper, they were able to get this clear distinction of blood cells from all of the others, and kind of a distinction of these stem cells from the others, but everything else is just kind of superimposed on everything else. So that was really interesting, that without doing any other work, we were able to get significantly better results than they were at separating these by anatomy.
Do you have any confounding factors with this data?
Confounding factors?
Yeah. Like, I don't know, male, female, like age.
So these are the experiments that have been performed by the roadmap project. So I know that there are different ages and there are different sexes, but I'm not sure.
So do you consider those things, or?
We don't really consider them because there's not much we can do about it. That they have, for this cell type, they have a specific sample. It's not that they have many different samples of them that we can choose from. So the ENCODE project now has a much more complete compendium of various cell types by different ages and sexes and so forth. So now let's take a look at some of the imputations that we get. That on the top we have the real data. This is one of our tracks that we held out in the test set. And on the top we have the predictions that are from compute, which is the first approach that came out, along with the mean squared error of those predictions in that region. On the back, in kind of transparent, we have what the real signal is in the back, so that you can kind of see what the overlap looks like. Then we have predicted, which is the approach that came out from our lab earlier. Then on the bottom, we have Avocado. So,
what's the scale here?
Whoops, the y-axis. So this is the arcsine transformed log 10 p-value.
No, the X scale.
Oh, the X scale? Wow, this is a terrible figure. Sorry, the x-axis here is kilobase, and so it's not 5. So this is 8 plus 1e whatever to the so it's basically kilobase. So there are some important insights here, despite how terrible this figure is. The first is that it looks like Chrome impute is, all of these approaches seem like they're kind of capturing what's going on fairly well. It seems like Chrome impute is over-predicting signal and predicting a wider signal than the other approaches. Like if you look here, you can see that there is this nice dip here that these other two approaches get, whereas Chrome impute doesn't really get it nearly as well. In contrast, predicted seems like it's getting there, but it's significantly under-predicting signal values across the whole genome. On the other hand, Avocado is perfect and everything is amazing. But more seriously, it seems like it's able to, it doesn't suffer from the problem of under-predicting like predicted does, though it seems like it might be missing some of these smaller peaks here, though all of the approaches seem like they are. So it seems like it's this nice middle ground between Chrome impute and predicted. So we then want to measure this across. We then want to measure this in a cross-validated setting across the full genome. So what we do is ignore this for a little bit. We measure this according to six metrics. The first is just the global mean squared error. How well are these various approaches, imputations, able to do at reconstructing the signal? And so you can see that predicted and Avocado are basically performing exactly the same, and both of them are beating Chrome impute. Then Chrome impute introduced this metric called MSE1 observed, which is if we consider only the top 1% of positions according to the observed signal in the roadmap compendium, how well do we do at those positions? Basically, these are anticipated to be real peaks. How well do we do at real peaks? And you can see that Chrome impute does incredibly well. They then gave this approach, this metric called 1 imputed, which is if we look at predicted peaks, what is the error at our predicted peaks? So it's giving a slightly different view. And we can see that Chrome impute does horribly, and that Avocado does the best. Then I added 3 new metrics which are based off biological interest rather than just a second. And so this is measuring the MSE at the promoter regions, gene bodies, and the enhancer regions. And in all 3 of these approaches, Avocado does better than the other ones and significantly better than Chrome impute. Yeah.
What do you mean by predicted peak?
So we are imputing the signal, and so if originally we were taking the top 1% of positions according to the actual signal, the run in the roadmap. But if we only look at the top 1 positions according to the predicted signal, the ones where you predict peaks, what is the error there with the real signal?
Well, that's kind of moving the bar a little bit in favor of your method, right?
It's not moving the bar, it's just a different view of what's going on.
I would disagree. Your method, naturally, because you're going to predict those to be strongest, that you are probably going to do best on those. So you should compare your predictions. So you should compare, say, Chrome impute's top 1% predicted peaks.
That's what I'm doing. This is the top 1% for each method.
Oh, okay.
Yeah, this isn't the top 1% according to my predictions. This is the top 1% according to each approach. So in Chrome impute's top 1%, what is the error in predicted's top 1% and my top 1%?
Okay.
Yeah, so now imagine the question marks come out. What is the discrepancy between the top 1% of observed ones and the top 1% of imputed ones? It seems like this is suggesting that when a peak exists in the real data, Chrome impute does really well at predicting it. However, they are predicting peaks that don't exist. And so my hypothesis this time was basically that these techniques are unlikely to just predict random peaks at areas. It's unlikely that they're just predicting random peaks where nothing exists. What's much more likely is that in areas that are peaks in some cell types and peaks in that are peaks in some cell types and not peaks in other cell types, that they are unable to distinguish which cell types the peak exists in. And so, to kind of make that more conceptually clear, what we want to do is we want to look at these, and instead of measuring the error into this, just according to the x-axis, what we want to do is we want to see we want to classify each position across all cell types. We want to say, for each position in each assay, how many times is this a peak? So here, these positions are peaks in all 3 assays. These positions are peaks in all 3 assays. Perhaps here, this is only a peak in 2 assays, and so forth. And what we want to do is instead measure for each assay, what is the mean squared error at positions that vary versus positions that are constant. So this is kind of a sophisticated question that took me a really long time to write down in a way that didn't confuse my advisor. And so what we end up with is a plot that looks like this for H3K4ME3. This is the mark that is localized in active promoters. So the x-axis here is when we look at positions that are a peak in this number of cell types, with peak being according to max2 that's run on the actual roadmap data, what is our, on the left-hand side, what is our mean squared error? And then in the middle, what is our vcall? Vcall measuring basically how well the signal localize in these, how well the signal localize in peaks, and precision being when we predict a peak, how good is it? And so both vcall, so mean squared error is just, we take our real value predictions and calculate the mean squared error between the real value predictions and the observed data. Vcall and precision are done when we binarize our predictions based on the threshold of log 10 p-value of 2, basically p-value of 0.01. This is the threshold that was used in the Chrome impute paper, and so we're replicating it. And this basically confirms our hypothesis on this type of data. What we're seeing is that Chrome impute has a very high recall, which means that when a peak exists, it is able to capture it. But it has a very low precision, which means. What are the colors? Oh. Okay. In cyan, the color cyan is Chrome impute, purple/magenta is predicted, and green is avocado. Is that clarified?
What's the fourth color?
Oh, blue is if we use the actual roadmap data. And the reason that it's not perfect for the precision in vcall is that when you call peaks, you typically have a region past where there's strong signal, because a peak starts at the base and comes to the summit, and so you're not going to have strong signal at every one of those positions. Okay, so to repeat, we see that Chrome impute has a very high vcall, which means that when a peak exists, it is able to capture it very well. It has a very low precision, which means that of the peaks that it predicts, not many of them are real. Whereas both predicted and Avocado do much better at that. And in fact, Avocado is able to balance the precision in vcall in such a manner that gives it the lowest mean squared error across as these vary. Yeah.
So for MSE figure, white drops, but the red tail doesn't mean top of figure?
I'm sorry?
White's on the MSE, the left hand of figure, white decreased as the more cells.
You can imagine that it's very easy for an algorithm to, at a position where it's never a peak, to say, hey, it's never a peak here. And then you're going to have a very low mean squared error. Likewise, when a peak always exists, it's very easy for one of these algorithms to say it always exists and have a lower, but not zero, MSE. It's really in the middle there, where sometimes this peak exists and sometimes it doesn't, that are the most interesting regions are going to correspond to the highest MSE, because you want to be able to get these, you want to predict which cell types these peaks exist in. Does that make sense?
But it's not possible, this peak shows everywhere in all the cell types, so it should be.
If a peak, so on the x-axis we have the number of cell types in which a peak exists in. And so if a peak exists in every cell type, what you see here is that it goes up and then it goes down again, because if a peak exists in many, many cell types, it's very easy to predict it's just going to happen there. But the area of primary interest is this middle region here, which is where the peaks occur in some cell types but not others, because those explain variability across biological cell types. We can talk more afterwards if it doesn't quite make sense.
Question over here.
Yeah.
It might be nice to know what the relative fractions of each sort of type of specificity you have, because I guess you're not normalizing this based on what's observed in terms of specificity of the peaks. Like, it's like, it would be good to know how many regions are found only in one cell type, two
Right.
cell, that may not be constant, right? So you have probably some distribution.
Yeah.
And you're not normalizing your performance based on that distribution, right?
It's true that we aren't normalizing it, but if we did normalize it, then we'd get the global mean squared error measurement. But it's true these aren't normalized, because you can imagine that there are going to be far fewer positions that are peaks in like 70 cell types than in 0.
Right.
But we're trying to focus on basically that's ultimately going to be a scaling factor, and it's not going to change the ordering of these, which is the most important one, saying that we don't care, for example, that the mean squared error is 0.75 here. We care that overall, it seems like Avocado is balancing the precision and recall in a way that provides a lower mean squared error than the other one.
But your performance could be based on how much information there is. Like if you have very few regions that are specific, then maybe that's not as predictable.
Sure, but that's okay. Yes, I think that would be interesting to include, and I don't have a figure here. Yeah.
Something similar. Do you use the same genomic windows?
Yeah. Basically, what we have is just long vectors that represent the VO data, the imputed data, and we're able to take slices and make comparable measurements across all of them.
So the Chrome impute, you use the same.
And so I'm going to kind of whiz through it because I have less than 5 minutes. But we see the same thing across DNAs, which is very different from the histomodification that we just saw. Avocado is better in other ways as well, but I'm going to ignore that. But the point here wasn't to do well at imputation. The point here was to learn an informative feature representation that can be used across a variety of genomics tasks that is broadly useful. And so what we did in order to evaluate it was basically the same type of procedure that I use when I order a hamburger in San Francisco. So step 1, choose a prediction task. And what we did here is that we evaluated predicting gene expression, promoter-enhancer interactions, frequently interacting regions, which are regions of three-dimensional structure that interact frequently, and TAD boundaries. Step 2, choose a cell type. And so that's very dependent on what cell types we have information for based on the task. Step 3, choose a feature set. And then the feature sets we have is whatever data we have available in the roadmap project, the full set of Chrome imputed marks, full set of predicted or Avocado imputed marks, the Avocado latent factors for the genome as the feature set, or the full roadmap compendium. So that's important that what we have is we have the epigenomic data that someone might initially use to make one of these predictions. We have all assays, imputed values, which is one way of fully characterizing the human epigenome. We have the Avocado latent factors, which are a competing approach for characterizing the human epigenome. And then since we are summarizing the full roadmap compendium, all 1,014 tracks, we just feed that into a model. Why not? And step 4, we run 5-fold cross-validation using gradient boosting on whatever cell type feature set task that we have. And so this was a lot of computational power, and we averaged the average precision recall. Sorry, the average area under the precision recall curve for each one of these 5 folds. So when we look at gene expression, the results aren't immediately clear. The gist is that the biggest finding that we found here is that when we use the full roadmap compendium, all 1,014 measurements, we do significantly better than if we use only the epigenomic data from a single cell type. This is not the standard way that most people do genomics tasks. This really surprised us when we did it. And the reasoning is kind of simple, actually, that using the full set of marks can give you cellular context for what's going on. If you know that like H3K4ME3 peaks in promoter regions and you see a peak by itself, then you might say, oh, this is a promoter in this cell type. But if you see that in every other cell type, it is not a peak, you're more likely to say this is noise. And a machine learning model can pick up on that. So that was really interesting. What we found is that the Avocado latent factors do better than that in 36 out of 47 cell types, and by an average of like 0.006 AUPR. And so what it basically seems like is that we have many cell types where this task is easy. And the Avocado latent factors in the full roadmap compendium kind of do equivalently well in that. When the Avocado latent factors start to shine is when in cell types that are difficult. These are potentially cell types that are not very well characterized or have particularly noisy measurements. You can see here that in these 7 cell types that are more difficult across the board, that Avocado does better by 0.03 area under the precision recall curve. And then we blow out just using the cell type measurements like one would typically do when making this, or when doing this task. Then, when we try to predict promoter-enhancer interactions, we use a gold standard set from the target finder paper, and we compare against the target finder method. And we use the same feature sets as before, except we include 2 feature sets that the target finder paper found useful, which is histone modifications and transcription factor binding and methylation from either the enhancer and the promoter or the enhancer, promoter, and window. And what you can see is that the Avocado latent factors do significantly better than the full roadmap compendium, like we saw was very powerful before, but also the target finder feature set that was derived from transcription factors and methylation. It's doing better than a feature set that includes information it was not exposed to. The Avocado feature set does not have any information about transcription factor binding, and it still outperforms it despite how important that is to doing things like predicting promoter-enhancer interactions. The only feature set it doesn't consistently beat is this feature set that includes measurement from this window between the enhancer and the promoter that Avocado doesn't have as well. Then we look at, I'm going to finish in just a minute or two. So the next task that we looked at was trying to predict frequently interacting regions, which are basically a subset of particularly active TADs that are enriched for things like super enhancers and SNPs of particular interest. And they were submitted by Bing Ren's lab at UCS State, I guess 2 years ago now. So what we find is a little bit of a more complicated story. That what we're seeing here is that when we use the set of measurements from before, that again, the Avocado latent factors are just blowing out of the water using the normal epigenomic data that one might do. But in this case, it's not doing as well as using the full roadmap compendium. And our hypothesis for why this is because we're doing this at 40 kilobase resolution, whereas the other tasks were at a much smaller resolution. Since the Avocado latent factors measure things that are like 25 base pair, 250 base pair resolution, and since they are Gaussian distributed, that when you aggregate them over such a large context, they potentially lose more information than using epigenomic data that is not normally distributed. So regardless, using the Avocado latent factors is significantly better than what people do, and it's over an order of magnitude faster than using the full roadmap compendium. And then we look at TAD boundaries, which we basically see the same thing, except more importantly, we still aren't able to, that everything seems like it's doing not that great at predicting TAD boundaries. This is a hypothesis that we were testing because people were not able to build very predictive models using epigenomic data, and we found that latent factors aren't suddenly able to make us do that. That we're not distilling this type of, we're not suddenly doing well at tasks that we weren't able to get any type of real predictive performance in before. So, I'm not going to talk about the feature attribution stuff, but at a very high level, the gist is that we have produced a low-dimensional latent representation that is able to succinctly summarize these high-level phenomena that are going on in the genome. In addition to doing significantly better at imputing all of this missing data, we also found that this representation is broadly useful for other genomics tasks that it was not trained for. And so sometimes dramatically better, like in the case of predicting promoter-enhancer interactions. So we're still in the process of trying to identify what types of tasks these latent representations can do well on, and what types of tasks that seem to be beaten out by the full roadmap compendium. But across all of the tasks, in all of the cell types, using the latent representation is better than using epigenomic data from a single cell type, like is the typical case. So I have other work that I plan on doing before I graduate, and you can read about it in my many papers that will come out at some point in the future, hopefully. And then I'd like to acknowledge the other co-authors, Tim, who is working on the predictive paper and has helped me significantly with understanding what biology even is. And then my advisors, Bill and Jeff, who do computational stuff. Thank you.
I'm sure some folks have to run off, but there is some time for questions for those who might want to remain.
Yeah.
You mentioned that one of the major limitations in this field is computing power. So, have you tested to see, the effect of the computing power on performance in your case? Like, have you tried different numbers of predictions? You have to mention like 1,000 trainings or something like that. So, whatever is like limiting, have you tried to see like what the minimal computing power is to get the performance?
Do you mean how much data would we need in order to get similar performance at making these imputations?
Well, no, not data, but just the modeling part. Like,
whatever iterations or whatever it is that you have to do.
Well, so what we did.
Because the number you looked at the number of neurons and number of layers.
Yeah, so presumably this kind of answers the question, which is when we make certain aspects of the model bigger or smaller, how does that affect performance? And what we found is that at the imputation task, it seemed like adding more factors to the, did not significantly change the number of the performance at the imputation task, whereas changing the neural network did. What we didn't do is then go and see whether or not, after learning a significantly smaller representation, perhaps instead of ultimately we had 110 genome factors, what happens if we have 2? We didn't end up doing that on the downstream task. Yeah.
But regarding the window size, you said you set the multiple window size, but I may have had this whole thing wrong, but the entire, they're not all the same total length, right? But then when you're sliding through each data set,
the data set is the same length. It is one measurement per base pair.
One species is all human.
This is all human.
Oh,
yeah, this is all human cell types. And so we have one measurement per position in the.
I don't know why for some reason I thought you maybe have more.
Well, one of our extensions is that we are trying to do this cross species because even though human is well characterized, other species aren't. And since these kinds of modifications operate basically the same way in other species as human, we can train our model in human, refine it for these other species, and then say impute all of them out into a project.
Okay.
But we don't do that yet.
Sorry, I thought.
Yeah. Yeah. It does take the mean. You mean for all the downstream tasks? No, so for every assay, we are aggregating a 25 base pair resolution. But in the downstream tasks that we have, these the ones at the end of the, what we do is that we are taking the average across whatever region of interest we have, like in the promoter and the enhancer, we're taking the average no matter how big that promoter or that enhancer is for each one of these feature sets. And so potentially there are more informative ways of representing that data, but we don't really consider that because we're just trying to demonstrate simply the broad utility of the latent representation instead of trying to build state-of-the-art models for each task. Any other questions? Well, thank you.